Hello everyone, I'm Satya, pursuing second MSc Microbiology. In this video, let's discuss about a fungal disease called rust of wheat caused by Puccinia triticina. Rust of wheat. Following the rice, wheat is the second most important cereal. It makes up to 20% of global food supply and the calorie needs of a population across numerous developing nations. Since the beginning of the years, wheat and rust have existed together, and rust have been observed to be the earliest known diseases of wheat. Puccinia triticina is the primary fungi causing leaf rust, which is a prevalent rust disease in wheat. Taxonomical classification of Puccinia triticina. Puccinia triticina belongs to kingdom fungi, phylum Basidiomycota, class Uridinomycetes, order Uridinales, family Puccinaceae, genus Puccinia, and species Triticina. History of wheat rust. In 17th century, the French farmers had noticed that black stem rust was much worse in the fields surrounded by hedges including barberry bushes. It was only in 1865 that a famous mycologist, Heinrich Anton de Berry, discovered the complete life cycle of a stem rust and demonstrated that Puccinia graminis required two different hosts, that are wheat and barberry bush, during the different stages of its development. The systematic investigations on cereal rust in India were initiated by Rai Bahadur, professor of Agra College, between 1922 to 1923. Professor Mehta's experiments have conclusively shown that barberry, an alternate host of wheat stemless pathogen, is not functionally involved in the fungus' continued existence in India. Wheat rust pathogens are obligate parasites which need living host. However, the wheat rust pathogens Puccinia graminis and Puccinia triticina were also grown on artificial media successfully. During culturing, mycelium grows but spore production doesn't occur. Epidemiology and geographic distribution. Wheat rust are known to survive and carry over to next generation with the help of iridospores, which are called as repeating spores. It is widely known that wheat rust survive on off-season, self-sown wheat plants in Himalayas and southern India. Some of the grasses growing in Yamuna Nagar of Haryana, Ropa district of Punjab, Una district of Himachal Pradesh, and area near river basins or other water sources may also act as collateral host for brown rust of wheat. Mountain barriers may account for the variation in geographic populations of Puccinia triticina in Central Asia. In South America, wheat leaf rust causes major yield losses in Argentina, Bolivia, Brazil, Chile, Paraguay, and Uruguay. In East and South Asia, high-risk regions for wheat leaf rust include China, India, Pakistan, Bangladesh, and Nepal, while in Central Asia, most of wheat crop is planted in areas prone to the disease. In Russia, leaf rust results in yield losses for both winter and spring wheat. In North Africa, leaf rust causes severe yield losses in Egypt and Tunisia. In South Africa, leaf rust epidemics frequently occur on the spring wheat in Western Cape, winter wheat in Orange Free State, and irrigated wheat in other provinces. In Australia, leaf rust is widely dispersed, occurring in all wheat growing regions. Host range of wheat rust pathogen. The primary host of Puccinia triticina include bread wheat, durum wheat, cultivated emmer wheat, wild emmer wheat, goat grass, and triticale. Puccinia triticina requires an alternate host to complete the sexual stages of its life cycle, which helps in evolution of new races or pathotypes through genetic recombination. Life cycle of Puccinia triticina. During the first stage, under stable environmental conditions, the wheat plant produces dark brown two-cell cheiliospores. In stage 2, Teliospores germinate to haploid basidiospores on wheat leaf tissue. In stage 3, the basidiospores are moved by wind and infect the alternate host to develop haploid pycnia. In fourth stage, the pycniospores are carried by insects to other pycnial infections, whereby sexual provocation between two genetically dissimilar cells takes place, resulting in the formation of plasmogamy. Finally, in the last stage, the acial cups release asiospores that will be transmitted by wind to infect wheat. The sexual stage of Puccinia triticina doesn't contribute to pathogen virulence diversity, hence reproduce asexually through the production of dikaryotic iridonospores. Temperature between 10 to 20 degrees and adequate moisture on leaf structure are conductive for infections. Depending on satisfying environmental conditions, the cycle is repeated every 8 to 14 days. The life cycle is complete when asiospores germinate and penetrate stomata of host, resulting in the production of asexual iridonospores. Symptoms of wheat rust. The symptoms include whitening of wheat spikelets or complete heads and dusty reddish-orange to reddish-brown spots that emerge on leaf surface. Countless spores are produced by these spots and they can almost completely coat the upper leaf surface. Water splashes and wind both disperse the spores of leaf rust. Both pictures indicate the symptoms of wheat rust. Reddish-brown spots observed on wheat leaf caused by Puccinia triticina. Management of wheat rust disease. Combinations of cultural, chemical, biological and genetic resistance can be employed to manage wheat leaf rust disease. However, genetic resistance is the most reliable, cheap and environment-friendly means of controlling wheat rust. Cultural practices. Leaf rust disease can be managed using cultural practices. The sources of resistance that already exist are further enhanced when a variety of cultural practices are implemented. Thus, crop management, which includes selecting the right crop, sowing it at right time and getting rid of weeds, may be able to effectively reduce leaf rust. Wheat leaf rust disease can be controlled by using fertilizers appropriately. Leaf rust can also be reduced by managing watering schedule. Crop rotation also reduces the amount of iridospores that get produced and deposited during the season and genetic diversity of pathogen population. Chemical control as an alternate strategy. Chemical methods of treating wheat rust become increasingly crucial. Control of leaf rust by chemical products is available with new and more effective fungicides such as picosystropin, flucosystropin, pyraclostrobin, propiconazole, prothioconazole, tebuconazole, metconazole along with pyraclostrobin, propiconazole along with azoxystrobin, fluoxapyrazide along with pyraclostrobin, prothioconazole along with trifluoxystrobin, tebuconazole along with trifluoxystrobin, and cyproconazole along with picosystropin. Biological control. The bacterial strain Pseudomonas putida have the capability to produce several types of antibiotics, siderophores, and slight quantity of hydrogen cyanide which suppress the Puccinia triticina growth in vitro and in vivo. Different biocontrol agents were evaluated against leaf rust of wheat, including Verticillium lecanii, Beweria bassiana, Pacillomyces fumosoroseus, Metarhizium anisophilae, and Cladosporium cladosporioides were used against Puccinia triticina in the field and lab conditions. Host plant resistance. Deployment of genetic resistance is the most effective, environmentally friendly, cost-effective and long-term strategy to control leaf rust disease for reducing yield losses. It is also the best strategy, particularly for poor farmers in the developing world. Host pathogen interaction could be specific or non-specific. Based on this, plant disease resistance can be classified into two major categories, namely race-specific resistance and race non-specific resistance. Race-specific resistance. Race-specific resistance is conferred by a single resistance gene and is also termed as qualitative, vertical, seedling, all-stage, monogenic and hypersensitive resistance. It is effective against only particular races of a pathogen. It is effective at the seedling stage and at adult stage. However, there are a few race-specific resistance genes that express their resistance genes at adult plant stage. For example, LR12, LR13 and LR22A. Race non-specific resistance. Race non-specific resistance is conferred by multiple genes and is also termed as quantitative, horizontal, adult plant, slow rusting, polygenic and durable resistance. It is effective at adult plant stages and is often detected as field resistance. It is also effective against a broad range of races and is an important source for durable resistance. Among the LR genes catalog, only 14 confer race non-specific resistance. For example, LR12, LR13, LR22, LR34, LR35, LR37, LR46, LR48, LR49, LR67, LR68, LR75 and LR77. However, there are exceptions where some race non-specific resistance genes provide race-specific resistance, that is LR13, or confer a hypersensitive response, that is LR48. Only a few wheat leaf rust race non-specific resistance genes confer partial resistance against multiple pathogens, and these include LR34, LR46 and LR67. Thank you for watching.